My name is Omer Berenfeld. I'm from the Center for Arrhythmia Research at the University of Michigan, and I'm going to talk about the role of fibrosis in atrial fibrillation. It has been known that fibrosis plays a major role in atrial fibrillation. We set our general objective to elucidate mechanisms by which fibrosis alters electrical impulse propagation and facilitates fibrillatory activity in three types of preparations and cases. The first one was in cell cultures, where we were studying the basic principles of propagation in fibrosis. The second was studying of the role of fibrosis during atrial fibrillation in a sheep model of congestive heart failure. And the third one was to study fibrosis in postoperative atrial fibrillation in patients. As for the first method, the myocyte fibroblast co-cultured monolayers, we used optical mapping to track the activation patterns, and we used numerical simulations of activation in two-dimensional models of ventricular myocytes and fibroblasts. This slide shows our cell cultures. They consisted of neonatal right ventricular myocytes and fibroblasts. The top left panel shows in green myofibroblasts and fibroblasts, and in red myocytes. The bottom left panel shows the confluency of our cell cultures, and here on the top right we see a bird-eye view of the monolayers that we studied, 35 millimeters in diameter. As we can see, the dark spots that correspond to the fibroblast indicate the fibrosis, diffusive patterns of fibrosis. We further looked at coupling between cells. It is not known if myofibroblasts and myocytes couple in vivo. We wanted to study our coupling in the cultures. On panel A, you can see that there are green spots between two myofibroblasts. On panel B, you see these green spots that correspond to Connexin 43 gap junctions between two myocytes, and panel D is a zoom in into the area between myocytes and myofibroblasts, and we can see here in green again gap junctions indicating a possible coupling between the two cell types. As for the modeling, for the numerical modeling, here we have the electrophysiological properties of the fibroblast. We followed the data from Shibakawa that studied the membrane current as a function of voltage in the fibroblast. We modeled those fibroblasts as non-excitable passive cells with high membrane resistance, and the resting potential was at about minus 15 millivolts. As for the coupling, it is not known what the coupling between myofibroblasts and myocytes is, so we modeled them as a scaling factor of the coupling between the myocytes. The specific question we asked is what is the direct effect of changes in fibroblast density and coupling on patterns of electrical activity. Here we see on the left snapshots from movies taken from the monolayers, and on the right snapshots from movies from numerical simulations. The colors in those panels are coding for the phases of the action potentials. Therefore, where all colors converge is a site of wave break, or as we call it, phase singularity point. What these snapshots tell us is that when we have 10% of fibroblast to myocytes area ratio, there is one single rotor. Here we see a clockwise rotor, and with one singularity point. As we increase the amount of fibroblast in the cell culture to 50%, the activity looks more complex, with more sites of color convergence that indicate more sites of wave breaks and singularity point. When we move to 70% fibroblast in the cell culture, there are indeed much more complexity to the patterns of activity with many more singularity points. So we quantified the number of singularity points in the experiments as a function of fibroblast to myocytes area ratio, and we could see a monotonic increase in the number of singularities, suggesting an increased complexity. When we went to the computer simulations, we see the same trend of increase in the number of singularity points with fibroblast to myocyte area ratio. However, in the simulations we did something which is very difficult to do in experiment, and that is to alter the coupling coefficient in the fibroblasts. What we can see is that when we increase the coupling from 0 to 0.2, the number of phase singularity increases at a later fibroblast to myocyte area ratio, suggesting that with more coupling we can sustain better organization of activity. We looked at the frequency of the rotation of the rotors we saw in the movies, and in the experiments we saw that rotation frequencies linearly decreases with fibroblast to myocyte area ratio. In the numerical simulation we saw that is also the case. There is a decrease of frequency of rotation with fibroblast to myocyte area ratio, and that decrease is independent of the coupling. Here we simulated 3 levels of couplings, all showing decrease in the in the rotation frequency. We looked at conduction velocity, and that also showed a decrease in value when we increased the fibroblast to myocyte area ratio, and that was also confirmed by numerical simulations independently of the coupling coefficient. We then moved to the whole organ studies. We looked at fibrosis and dynamics of propagation in sheep failing hearts during atrial fibrillation. We tachypaced the right ventricle of a sheep for 6 to 8 weeks to generate heart failure. We then isolated the hearts, and we optically mapped the posterior wall of the left atrium in isolated hearts during the induced sustained atrial fibrillation in the presence of acetylcholine, and we further stained sections of the atria for spatial distribution of fibrosis. Here are data from frequency analysis. We first see in panel A the area that we mapped. That area consisted of the posterior wall of the left atrium as exposed here. In this picture. Panels C and D are showing us the frequency maps of control and heart failure. We can see based on the color code that the frequency in point A is about 19 hertz, and in point D it's 6.8 hertz. When we looked at the cumulative data, we can see that the dominant frequency, the maximal dominant frequency in the various regions form hierarchical organization. In control, the posterior left atrium was the fastest as compared to the Bachmann's bundle and the right atrial appendage. That hierarchy remained but was blunted in heart failure, as seen by the red bars. The frequency was significantly lower in the heart failure as compared to control, but nevertheless was faster than the Bachmann's bundle and the right atrial appendage, indicating that in both cases, in control and in heart failure, the posterior left atrium is probably the site most likely to harbor drivers that maintain atrial fibrillation. We looked at activation patterns during atrial fibrillation in control and in heart failure. The top set of snapshots indicate sequential activation in the posterior left atrium. In control, we see that the site of breakthrough, this is a point of initial activation of the posterior wall, and we see outward activity from that point, and the site of origin is the same in those 4 sequential activations. However, in failing heart, the patterns change from beat to beat. Here we see site of activation, original activation in the top right side of the posterior wall, and then the next activation started more to the left and to the bottom part, and that changed in the 3rd and the 4th activation. We quantified the number of changes per beats per second, and we concluded that in heart failure we have more changes of beat to beat patterns of activation, indicating less stability of activity during in an AF of failing hearts. We looked at that in more details, and we first noticed that most of the activity that was seen during the atrial fibrillation in those hearts were of breakthrough types. However, there was a difference between the control and failing hearts. As for the outward breakthrough, those breakthroughs that going outside of our field of mapping, most of them were seen in control hearts. However, for the inward breakthroughs, those that are propagating mostly towards the center of the posterior left atrium, posterior wall of the left atrium, most of them were in failing heart. This is beautifully demonstrated in these diagrams. Each point corresponds to a site of breakthrough, and each different colors correspond to different experiments. And they were all placed in that diagram of the posterior wall with 4 pulmonary veins. But we see that in control, the sites of breakthrough tend to be centered in the posterior left atrium, while in failing heart, those breakthrough sites seems to be dispersed towards the periphery of the posterior left atrium. So we put forward the hypothesis that the reduced frequency and stability of activation, as well as the shift in the breakthroughs, correlate with the spatial distribution of fibrosis. And here we have stained sections from the center in control heart, center of the posterior left atrium, and in the periphery in control hearts. We can see that there is slight fibrosis in both center and periphery. However, there was no significant difference between the center and the periphery in the amount of fibrosis. However, in failing hearts, both in the center and the periphery, we see a much larger amount of fibrosis compared to control. We looked at the percentage of fibrosis in all the hearts, in 4 hearts, 4 control and 4 failing hearts, and we noticed that in failing hearts there is much more fibrosis as compared to control. If we looked at the architecture of the fibrosis, we see that as for diffuse type of fibrosis, there is more control hearts consisting of that type, and as for the patchy fibrosis, there is more in failing hearts. And this was further demonstrated by looking at the size of the obstacle. If we look at control hearts, both of them have the same size of obstacle, of fibrotic obstacle. However, for the heart failure, we can see that there is an increase in the obstacle areas in the periphery of the posterior wall. And this is also indicated by this histogram of the number of obstacles versus the size or the area of each of the obstacles. We can see these red bars corresponds to large obstacles that are exclusively found in heart failure. We then move to patients. This is the third approach here, this study. We looked at patients post-cardiac surgery that were instrumented with left atrium, right atrium, and right ventricular bipolar leads. We then performed spectral analysis of the recordings, and we also stained for fibrosis biopsies obtained from the left and right atrium. This is a picture of the open chest view of the heart. This is the left atrial appendage, and here we see the wires that are connected to the left atrium, right atrium, and right ventricle. In this slide, we see that in a schematic, we have wires to the right atrium that are creating bipolars that are not so closely located. This is because of limitation posed by the procedure itself. Here we have left atrium wires, and we have 2 wires in the right ventricles. We looked at 44 patients for biopsies, and 27 patients in which we placed the leads for recordings. Of those, 9 patients had atrial fibrillation, and 17 remained in sinus rhythm. And of those 9 that were in AF, we analyzed 7 patients in which atrial fibrillation lasted more than 15 minutes. This is the population characteristics. As we mentioned earlier, 12 developed atrial fibrillation and 31 remained in sinus rhythm. This is more or less the percentage of cases, 30 to 40% of patients in atrial fibrillation in post-operative follow-up. 30 or 40% present develop atrial fibrillation. The main differences between these two groups is that those that develop atrial fibrillation are usually older, they have more diastolic dysfunction, and with regard to heart failure, the situation is not so clear. We saw that most of them, those that remain in sinus rhythm were in class 1 heart failure. However, we analyzed, of those that we analyzed, 6 out of 7 were in various degrees of heart failure. As for the procedure, the heart surgery procedure, there is no clear indication of what type of surgery promotes atrial fibrillation. However, we noticed that 21 patients that went bypass surgery remained in sinus rhythm, as compared to 4 patients that underwent bypass surgery and developed atrial fibrillation. Here we show the frequency analysis of recordings from the left atrium and the right atrium. We see on top the time series that were recorded simultaneously. The ventricular contamination was removed, and here we see the power spectrum of the left and the power spectrum of the right. Noticeably, the dominant frequency in the left is 6.1 Hertz, and in the right atrium is about 4.4 Hertz. Cumulative data shows clearly that the dominant frequency in the left atrium is higher than the dominant frequency of the right atrium, and this was held constantly for 1 hour of recordings. We then looked at fibrosis in those patients. On the left, we see stained sections in a patient that remained in sinus rhythm. This is a young patient of 30 years old. We see no fibrosis whatsoever in these panels. On the right, we see stained section from a patient who developed atrial fibrillation. This is a female of 70 years old, and we can see that she had a 16% fibrosis, area of fibrosis from analysis of these sections as shown in panel D. We looked at the percentage of fibrosis in comparison with the development of atrial fibrillation, and what we found is that in those patients who remained in sinus rhythm, there was no difference between the amount of fibrosis in the left and the right, which was a little bit less than 5% overall. However, in those patients who developed atrial fibrillation, there was a significant increase in the amount of fibrosis in the left atrium, on average about 10 or 11%. We then, of course, looked at the relation of the amount of fibrosis to the frequency of activity to compare our results with previous approaches in animals and cell cultures. And we looked at the dominant frequency in the left atrium, which was the fastest side, and we noticed that the dominant frequency decreases as the percentage of the left atrial fibrosis increases. And we looked further at the gradient between left atrium and right atrium dominant frequency, as this parameter was found to be important in guidance for ablation. And we noticed that that gradient also drops with increase in percentage of left atrial fibrosis. So finally, here's a summary and conclusions. In co-cultures, increasing fibroblast to myocytes area ratios reduces the frequency of reentry and conduction velocity while increasing the overall complexity of conduction. Increasing fibroblast to myocyte coupling supports electrical activity at higher ratios of myofibroblast infiltration. During atrial fibrillation in failing sheep hearts and patients post-cardiac surgery, the increased fibrosis is associated with reduced frequency of activation, and heterogeneous spatial distribution of fibrosis governs patterns of propagation. Finally, I would like to acknowledge the many investigators that contributed to these studies and the support we obtained for various agencies. Thank you very much.